So I'm Walter Jetz. I'm a faculty here, a professor in ecology and evolutionary biology, as well as the School of the Environment. And thanks for joining us here in New Haven this week. It's a good time of the year to come here. I hope you enjoy the scenery. Feel free to grab any one of us, also about other aspects of life and enjoying the town here. I'm grateful to first of all NASA for the work and the agencies that joined up with NASA to support this larger activity. Keith Guys just joined us here from headquarters, I'm grateful also to, of course, our Max Planck partners who've been, as you know, been developing some initial pioneering technology here, not the very initial ones, but very recently, some sort of eye-opening proof of concept work. And in my talk now, I'm mostly just going to double down on many of the things that Ryan already mentioned, and it's sort of largely it's visionary, just thinking where could this take us. I'll give a few snapshots of some work that has happened here and trying to develop some of these foundations as part of the Max Planck Yale Center for Biodiversity Movement and Global Change over the last years. And highlight also some select work, some members of the center here. But several of them will be speaking later, and then we'll be able to double down further and provide some of the important specifics. So just a brief reminder, I think Ryan made an incredibly compelling overall case to the choir here about the importance of biodiversity, but just a reminder of this being also very important year that we will be referring to again and again in the coming years as 22, the year when there was an important IPCC meeting that's starting next week in Egypt, and in 2 months there is a very, very, very important convention of the parties, 15 meeting of the convention of biology. This is a COP15 coming up, and these are both 92 Rio conventions that obviously are intersecting with one another, and something that we will, I hope, be talking about in the coming days here and in the upcoming talks is that yes, animals as a way, and animal tracking as a way to keep a finger on the pulse of life and the health of the planet, but also as ways to understand the climate, and the climate is changing, how the environment is changing broadly, and potentially be sentinels of that and sensors of that in both ways. So we'll be getting into that a little bit as well. So I think there's the animal tracking does really intersect both of these areas, specifically as increasingly we'll see more and more onboard sensors of the conditions that can be linked up to the behavior of the animal that's carrying that signal. So it's been exciting to begin to work with data sets that, like MODIS, that obviously have been worked on a lot over many years, but now we've begin to work on something that's a 20 year stack of data, right? And it's something extremely rich. So these satellites have been up now for 20 years. Landsat has been up for longer, and we are able to use even some of these older products in new ways now because of that long-term data type that they're offering to understand not just change, but also to understand in more detail the biodiversity relevant variables and conditions, such as, for example, cloud cover, which turns out to be a much better predictor of many ecological attributes, biodiversity attributes, than precipitation, for example, or other issues. As Ryan said, there's a whole slew of interesting new satellites and products arising. I'm not going to get into the specifics here, but the important point for us, for our community, is that there's more and more products that have an increasingly strong biodiversity relevance. So some of these types of information, such as primary productivity or so, there have been other satellite products up there, but they've been very coarse grained, like 100 kilometer, 30 kilometer, and they're now moving to a spatial resolution where it gets interesting for actual ecological processes, even animal behavior. Same evapotranspiration. JEDIA is giving a very 30-meter pixel vegetation structure information that you can then link with remote sensing and some of these other products, Landsat, Sentinel, Modus, to then interpolate among the places where it's sampling to get to canopy height, canopy structure layers that are contiguous, and alongside immense progress around plant functional traits, also the surface biology geology mission coming up in several years, that, by the way, also came about partly due to sort of a community process, right? I remember sitting in a workshop at NC's in Santa Barbara 8 years ago or something like that, right? Where scientists essentially from different communities said, well, no, we should really have plant functional traits. Imaging spectroscopy, hyperspectral imagery is really, really, really powerful for this, for that, in ecology and biodiversity science. And so they are the remote sensors, NASA folks, the larger community, they are invited in biodiversity scientists, which was already happening at the time, but is more happening now. And it was an important moment to have these conversations and to together then be able to make a case that this should be something that shouldn't just be sitting on planes that are limited in where they can go, how far they can go, but indeed that should become a proper satellite mission. And that went into the decadal survey. And now it's a mission that's we're aiming for 27, 28 launch, if I'm not mistaken, that is going to deliver incredibly powerful and biodiversity relevant information at 30-meter resolution. So this process isn't fast, but it works and it does deliver. So I think this is something that you should keep in mind here, and I'm really grateful to Ryan and his leadership, as well as the leadership of others at NASA headquarters, to support us taking some steps in that direction. You all know that GPS tracking data has been already exploding, as we were saying earlier, right, through a variety of tags and solutions, many of them cell phone, SIM card based or Sigfox and other types of technology. And generally, data are exploding. Spatiotemporal occurrence information is exploding altogether, and movement data, by the way, is taking up a larger and larger portion of it. Doesn't mean that it's necessarily already that useful for ecological monitoring globally, because obviously it's limited to just a subset of species, but as we'll be talking about here, that is also changing. Let me frame this growth of spatiotemporal occurrence data and remote sensing information that's increasingly rich and biodiversity relevant in a way that was developed under the auspices of the Geobiodiversity Observation Network. GEO brings together the Earth observation communities across the world. It's an international organization, and then within it there is the Biodiversity Observation Network. And within that, there is a group of us looking after species populations working group. And as part of that group, also in the multi-year process, we began to conceptualize something that we called the essential biodiversity variable for species distributions and species abundance. And some of you might be familiar with the essential climate variables that really helped concretize some of the activities that the IPCC did. And in parallel now on the GeoBond, we've been developing essential biodiversity variables. So what that is something where we move away from these occurrence records that in some cases might also be including absences that tend to be sparse and biased in space and taxonomically as well, by using environmental layers and using that and bringing in model-based approaches that then let us fill in species space-time cubes. So here we have time, like year or month, and then here we have space, where each of these would be a pixel in space that belong to this country versus that country, for example. And that would then allow you to fill in that whole space across a large set of species. And then with this, you actually have that full coverage in space and across time that would then allow you to something that does have this policy relevance with which I started, right? The larger global policy frameworks, and that is indicators, right? It's to get from raw data to indicators, you got to do something in the middle to really be able to speak to a whole region, a whole country, and do so comparatively across all countries of the world, for example. And obviously, we are far distance away to do that with GPS tracking information. But I think we shouldn't be hesitant to envision something like this to also be done or partly supported by GPS tracking data. Many of us are sitting more in the wildlife management arena, right? We think about 10, 20, 30 species, maybe just 2 species, and they are really, the tracking data is really helpful to help us understand what's going on in the ecosystem, or how we can better support these species and all of that. But I see a really exciting dimension, or a partial dimension of what we're able to do in this 5-year effort around envisioning, okay, how could we really think about packaging this to become a multi-species indicator of sorts, indicator of connectivity, of land cover change, of the health of ecosystems, etc., etc. So you're going to abstract here. You'll have to abstract I think multi-species type situation, right? But that's in a way over the last year, sort of our eyes got open, or through that animation that Ryan showed, that actually is based on true data. We can begin to think about this. And then here, so how did we envision this at the time? So for species distributions globally, so not the animal movement at all, but there we saw initiatives such as Map of Life as something that can be really helpful in the middle here to provide a platform for engagement and model-based data integration. Down here, there are already central players like the Global Biodiversity Information Facility. MoveBank, of course, would be the place where a lot of movement data is stored, but the really important thing is that the data is coming together in some form, that it has metadata, that it's shared, truly shared, and can then be used in combination with other data sets, right? And in combination with remote sensing data to do integration, synthesis, that allows you to then deliver products. And that's really quite central. So there's the raw data side, bringing that together, the sharing, the incentives, the attribution, of course, for sharing, and then infrastructure efforts that help in the middle here to develop something like this. For example, if we want to address ultimately a larger scale indicators and use it for a larger scale monitoring of biodiversity or indeed climate, which is not all of the use cases we'll be talking about, but just a brief look at this one. And then there's obviously a whole infrastructure side here in the middle aspect, and then there's various communities that are engaging. This is very generally around species distribution data, species distribution modeling, that sort of stuff, right? But I'm showing this, even though it's not perfectly relevant to movement data necessarily, in that what I think is relevant for movement data, that we'll also need to think about these general aspects, right? The data integration and reporting aspect, the product production aspect, right? And the then downstream getting it out there to users, because that's ultimately why NASA would be supporting, or the US government would say, yes, we're supporting this, right? Because there's got to be some downstream larger use than just referring to a few species or a few national parks. So that just is a little background from a related, larger umbrella community. But one thing that is actually relevant for the movement side of things is this aspect of fusion, of data integration, and with that I mean data integration, not different movement data, but from different species and different places, but data integration from different domains. Remote sensing and climate data, and animal data, or species data, in our case, animal data. And there has been already really dramatic progress in that arena to do that intersection or infusion, or what's also called in the community that I'm spending a lot of time in, annotation, right? Essentially you have biodiversity data, an occurrence of an animal in space and time, and then you would environmentally annotate that. And many of you are familiar with that. Some of you run species distributions model, that's what you do. You do that in R, you pull out down a layer, and then you grab the data from that layer that connects to your points. In MoveBank, you might be using EnvDat. That supports some of that connection. What we tried to do also with earlier NASA funding out of the AIST program, develop a more general solution that really thinks about this as well, the issue of needing to and wanting to annotate biodiversity data in a flexible dynamic, and most importantly, scalable way. So that whatever is done here works for different data types, whether that's the museum records, citizen science observations, or indeed movement data or camera trap data. And the reason why you want to be flexible here and have a solution that allows, for example, to bring in different resolutions, with that I mean grains of spatial or temporal data, is because your data itself will vary in its grain and in its uncertainty. So if you work with camera trap data, usually you have a GPS location for that camera, you have very, very detailed, highly precise, highly certain data in space and time. Other biodiversity data, which is important as we want to generalize in the way that I was talking earlier, right, might be much coarser in its spatial grain, like a national park checklist, for example. We know something occurs here in this. In the US, we know much better usually, but if you think about the tropics, etc., that's sometimes all you have, right? And if you want to link in other species, besides the one that you have particularly good, precise data for, that might be the grain that you're stuck in. And then there is uncertainty. Usually it's quite small for GPS tracking data, but also not to be underappreciated, right? As we like with Planet or other types of remote sensing, we go to 50 centimeter or meter resolution. And we work with animals where the GPS sensors might be under the canopy or something like that. The uncertainties can actually be in the tens of meters. And we need to be mindful about that issue. And then in specimen records, museum specimen records, obviously, several years ago, we did an analysis of what is this rate of the data that's been shared in GBIF. What is the average spatial uncertainty for mammal data? That was before movement data was in there, et cetera, et cetera. And it was, for a particular snapshot at the time, it was over 10 kilometers, the spatial uncertainty. Right? And obviously that is really problematic as you want to link that to 10 meter, 100 meter resolution consistent data. But it's something you can work with if you're then also adjusting your intersection, your annotation in that same way. You bring along not just the 10 meter pixel intersection, but also the surroundings, which could be the ones that actually refer to that point, which is not a point, but more like this cylinder. And then mechanisms may also obviously depend on the spatial or temporal extent. So why a particular animal is here, growing here, or spending time here might have to do with the larger landscape situation rather than what's specific about this particular place, right? So you want to think about the environment, maybe in a kilometer buffer, 10 kilometer buffer, depending on the size of the animal, the home range, and all of that. And temporal drivers as well, right? So the rainfall in the week of where the observation came from might not matter so much, but it might matter from the 6 months before, right? Or the 2 years before, etc. So this is obviously no news to you, but what I'm trying to make the point for is that what we've been missing so far is actually a straightforward, easy, scalable way of doing these sorts of environmental characterizations that would allow you to have the flexibility and process relevance that you might have. So we prototyped, or actually developed, published, and made available this spatiotemporal observation annotation tool that we ran for across about almost 1 billion records for terrestrial vertebrates, as they had been shared by GBIF about a year ago. And that allows you to then look at what we call the species environmental niche as provided by the points where the observations are coming from in a very flexible way. So you can look at Landsat 8 and EVI at the spatial resolution of 100 meters and the temporal resolution of 30 days and look at what that looks like. And relate that to the CHELSA, which is a model-based precipitation layer at 5 kilometer resolution and temporal resolution of 1 day. And then look at that, right? And that would be sort of a niche. And you've seen these ellipses, right, that characterizes this niche. But there's many ways in which you can develop this ellipse, right? Even for a single variable, depending on the spatial and temporal grain that you use, or how much you're accounting for the precision and the uncertainty that goes along with the data that's on the changes. So this is sort of a versatile tool that you can play with. Here we have, for that species, the ruby-throated hummingbird that left here again about a month ago. So that's a species that migrates, spends time up here and then migrates. 700,000 records, etc., etc. So you can then begin to characterize the niche of that species in quite some detail and with quite some versatility. And then just think movement data. So for that species, there isn't any movement data in there, it's too small, but for other species, there might already be some movement records in there. And the way we built this, obviously, is that it's meant to support exactly also movement data. And then you can go beyond thinking about the whole species, but you can think about single populations or single individuals, and we'll be talking more about that. Here, an assessment for that species of MODIS line surface temperature for the day of observation here across since 2013 or something like that every year. And obviously that species migrates and then it disappears. Essentially, we don't have many records from the winter location, right? These are mostly citizen science-based records either. It would get into this. Do I need to finish? No. Okay. And you see they're keeping a pretty tight temperature space. And then here you can look at where that species is sitting in relation to all the hummingbirds, all the hummingbird data. Sorry, the species is here. Other hummingbirds have a wider range of maintenance temperatures. Based on, I don't know how many millions, tens of millions of points for all hummingbirds. So this is then something that's available for a whole range of different variables, Landsat, MODIS LST, precipitation, Jetz, MODIS, NDVI, etc., and different spatial and temporal resolutions. And you can immediately look at the correlogram, right, and see, okay, which ones of these characterizations are very similar with one another for the particular species and which ones are more different. So you can stitch together which combination of variables might be best to characterize that niche of the species. And this is still early days, so that needs another decision support tool on top. Ideally, that would help you do this automatically, maybe machine learning supported, pull the best predictors together and go from there to then have the best possible foundation for assessing the baseline niche of that species, and then you can relate it to climate change, etc., etc., etc. So one point here is that this is now technologically sort of doable or done in the first instantiation, and it begins to offer us a lens into not just the spatial distribution of species or individuals, but also their distribution in environment space, flexibly developed environment space. And that is something that's really powerful. It allows us to appreciate for this species that's mostly hanging out in winter where it is. We might bump into it here in the parks. The titmouse. So it's just swinging with the environment, right? In the temperature that it has. So it gets cold. It has it cold sometimes of the year and it has it warm enough times of the year. So these need a transformation to really get to degrees Celsius. I've been tripped up there. But it's a seasonal species that deals with the seasonality by not migrating and staying on and having adaptation. So this is the ruby-throated hummingbird again. And by the way, all of these you can look at a map of life and then appreciate how these conditions then play out in space. So this species keeps a very constrained temperature space. Now here the white stork, where we do have movement data, that's already showing up here. So these are citizen science records, some museum records. White stork, of course, one of the key species which we started to appreciate. Animal migrations, right? You know the image of the photo of the white stork with the arrow in the wing, right? That came back from Africa. So this is an enigmatic species and has been a really central model system actually for some of that large-scale thinking around what could GPS tracking deliver for ecology, for a large-scale niche understanding, for a large-scale ecology and conservation broadly, because it's large enough that there have been tags on this species for quite a while. And because it's so, people really care about it, it's been tagged throughout. And from Ben Carlson, we'll hear a presentation where he's specifically going to talk about this. But here, a mix of citizen science data and then already moving animals here. So these are mostly, I think, almost exclusively GPS-tracked individuals. We humans, didn't appreciate how much storks are hanging out in this area. Actually, the local people appreciate it. But here, our scientists, who unfortunately are mostly sitting up here or here, didn't, and they actually spent long weeks there, mostly going after emerging insects that are a plague there. Yes. Thank you. So they are really tuning in on this, and then some of them from here that further. Anyway, you're starting to appreciate why this blue was there, right? Why the expert drew a blue here? Well, this is a wintering range. You know these from field guides, right? But now we get really the actual dynamics of this in a powerful way. And then we can look at this in niche space as well, right? Across the years, etc., etc. And this is not totally new, right? Some of this has already been done. Andrea Flack published a paper on some of this, on how some of these individuals are keeping their niche as they migrate and all of this. But the exciting, I think, news here now is that we're able to do this at scale, and we can flexibly characterize these things. It's not just something that you do, like you push a button in InfDAT and then it produces something for that individual. We can begin to offer this up already in a way as GPS tracking data comes in and is shared. We have workflows so that you can immediately look at this and share the niche perspective as well as the spatial perspective. And then here for that species, just showing you different grains of which we characterize the greenness of the vegetation here with notice. They're closely related, but not identical. And then here a brief shout out to Ben and his work, where he then moved looked at this not at the species level, but at the single individual level with GPS tracking data. But the same idea that we use movement tracks, environmental variables, to then estimate something called a hypervolume that actually in some of its key conceptual developments happened right here in the building where you had coffee, where Hutchinson had his office. Yeah? He's one of the key developers and pioneers of the concept of the multivariate niche, the n-dimensional niche, and he was on the faculty here now, department. So you can now do this. You can re-envision this old, long-established concept of the niche in new ways with movement data at the level of single individuals, in a way that allows the combination of animal behavior and ecology that I'm sure somebody like Hutchinson would have dreamed of at the time to be able to do. To have the single individual components here and be able to aggregate from there. And what Ben did is he pulled out these types of environmental layers. Some of these are seasonally varying, changing all the time. Some of them are static, 16-day distance interval, that would be static. But all of these are quite high resolution to then develop niche characterizations that, for example, in non-metric dimensional space could be characterized like this, where then each of these would be a single individual that we see here in the space. These are its home ranges. But you can immediately think about them also in niche space. So that's one thing that I find quite transformative in this arena, that we can now quite fluidly go back and forth between geographic and niche space. You can think about those simultaneously. There's more work to be done to really make this fluid, but it's I think the key proof of concept and initial attempts are there. And hopefully that's something we can bring back into some of the conversations that we're having this week. We as part of a workshop that we held here at Yale under the Max Planck Yale Center 2-3 years ago now, just before COVID, I think, we developed some concepts. Costa Pereira, Brett Chesner, and Remington Moll were the key people who then helped advance this further. This was just a perspective piece that thought specifically about community ecology as an arena that might benefit in the future. Not already, but looking ahead, the fact that we have multiple individuals, sometimes from multiple species, tagged in the same landscape. So that was the key conversation that we had here a few years ago, this multi-species, single-site capture of movements. Some of that you saw in that animation that Ryan shared earlier, right? You saw the storks coming together with the buffaloes in this place, right? And there are even some, actually, we know there are interactions between the two. You can actually see storks walking behind them and grabbing the prey, right? That they disturb through the buffaloes disturb for walking around. So we know there is interactions that we're really able to see and capture with GPS tracking, and more of this will come. Kruger National Park is a place where I know there's a lot of GPS tracking happening already across multiple species. Impala in Kenya is another, as well as places in the US, right? There's some outstanding systems of 2 species, 3 species being tracked simultaneously. You know exactly what this means, but the more this can be scaled up, the more we can think about this benefiting community ecology broadly, right, even across trophic levels. And we can, alongside, bring increasingly higher resolution environmental layers into the picture to then think about what we, in a simplifying way, called Eltonian factors and capture them. And that wouldn't be including the environmental part yet, but here we simply are able to interpret these movement tracks in a way that might signal and deliver us information about biotic interactions, right? So as these are coming together in the same, this would be pairwise spatial proximity, right? So as this one comes here, it might encounter some aggression. This one gets predated upon. This one might have a mating going on between two individuals, right? So this is all being delivered. So these are always two individuals that are characterized by a single line, just to clarify, right? When they're coming together, some stuff happens in the overlap in space and time, right? Could be mating, could be death, it could be aggression, or it could be just a happy stork walking behind the buffalo. Some so these we call Eltonian factors, and that allow you ultimately, you could think this up as finally, you could build this up to one sort of interaction topology, right, with enough data. This is almost a bit visionary. And then this part here, the environmental data would allow it to get at the Grinnellian factors. So Grinnell in 1917 is on the faculty at Berkeley, published the defining paper on the environmental niche that we're still talking about. So Hutchinson refined that concept later as an n-dimensional hypervolume, etc., and made some other important tweaks to the idea. But ultimately, in this sort of pure realized environmental niche perspective, Grinnell was the first one to say, well, we can think about, from where the species occurs in geographic space, we can think about the environmental conditions it requires, we can characterize these, and we can put them back on the map, right? That's what kind of what he did at the time. So we can also look at these single individuals and their individual environmental associations. And as we know, and as is already being characterized, there's quite a substantial among individual variation in how individuals use the landscape, their personalities, not just in how they behave with others and interact with others of the same species, but also in how they use the environment. And that's something that you can also characterize over time, how they differently use different parts of the environment. And then you can calculate environmental similarities from there, right? And assess how much they're all doing using the exact same environment, might be nested or might be overlapping or not overlapping, etc. So that's what I showed you earlier in that white stork example that Ben will be talking about. It's the same characterization, right? How do the individuals not just use different parts of geographic space, but maybe also use different parts of environment space, or use the same environment space, but in different parts of geographic space. And that, as you might imagine, has immense relevance of how these particular individuals and the species that they contribute to, that they're a part of, will fare under changing conditions. Right? As climate changes and moves these environments around, the way individuals are doing all the same or might have lasting or genetically defined different environmental associations will drive how climate change will affect that species' survival, population survival. And then across a large, multiple communities, so we're now doing comparative community ecology, you can do the same thing and characterize them multiple species and see how different populations, communities ultimately play out differently here. So this is anyway something we can now begin to envision. Like 5 years ago, we wouldn't have dared to draw these things, but thanks to sort of this excitement around Icarus, multi-species data now becoming more and more commonplace. Many of the demonstrations, the publications that many of you have done in the literature, right, of showing multiple species tracked in one place and what that gives you. We're beginning to be able to dare to envision something like this. And then this was also a daring, but also in this case empirically supported map. With the one pity being that after this map, then unfortunately the tracks stopped. This is the first large batch of data that came from Icarus, so that's about, I think, 6 months or so of data collection at that time. This was put together by the Max Planck Institute of Animal Behavior, that particular graph, and they have been obviously working with all these different individual partners and been helping them with these tags that were being read and might still be being read out by the Icarus antenna and system on the space station. And I know at least a couple of you, one or two of you are engaged in some of these species and have been doing this work. But what was eye-opening about this is that, wow, we can now look at this at the global scale. Not that we didn't already have movement data from all of these places of sorts, but in some cases, it was eye-opening because not only did we get these large-scale migration tracks that we already often had before, but even for small species like blackbirds, that was already something quite impressive in its own right. But additionally, we would get these local pieces of information, right, about the local home range, how they were really using the environment in square meter resolution for a long period before they then went on to thousands of kilometers of movement. So that was kind of, I think, a really important moment in time to like, wow, we can think about this as a planetary system, right? And begin to think about animals not just telling us about large distance connectivity and migratory patterns, but also the same thing about fine-scale use of the habitat and the environment. And as you think about this in a changing environment, right, like, wow, what that's going to be. And the powerful thing about this was also that it was all this, the data had the same data structure, it came from one system, it was all together, you could immediately begin to analyze it. And obviously, these are all tied to individual projects, and there was an embargo, obviously associated with that for just throwing it all together and displaying it like this. You got permissions to do this in this visual way. But you could begin to think about it in an abstract way, well, putting this together and either after the embargo or in a simplified abstract way already before, just to get some sort of overall signal, right, of what might be changing, where is it changing, what that might be associated. And away from the single species identity as a larger biological Earth observation system, or a way to have sentinels of biodiversity change. And then much of our thinking at the time, well, now we got to raise the funding to get enough tags, so we really can do this globally across many species. So we came up with this 100,000 sentinels idea that we were beginning to fundraise for, but then obviously, as you know, the Ukraine war happened and the Russians stopped the data sharing from the space station. They might still be collecting that data, hopefully someday, for species, individuals that are still flying around with tags on them. Their data will be usable for research, but not now. And there is sort of regrouping, and Martin will be talking more about that. But it was eye-opening, right? Because it allowed us to now more concretely think about this vision, where obviously that was already very well developed by Martin Kelsky, by many others, including many of you, that yes, it will be so awesome to be able to do this multi-individual capture of movements in a way that allows us to go to smaller, to more species because of these smaller tags, 5 gram, 4 gram, 3 gram, and to then combine that for community ecology, to look at spatial patterns, etc., etc. And several of us have already been thinking about the environmental dimension of that as well. And then at scale, you really will be able to begin to think about this. Ties back again to the initial slides that I showed, like this indicator idea. The idea that this would help us through models then, and maybe in combination with other data types, get a movement data informed characterization of how the landscapes are changing, in terms of how they're useful for the animals that have tags on them, or that might be representing certain traits, certain functions, or how the environment is changing because the sensors that these animals have on them, or what we know about them and their climatic niches, the animals are telling us directly about that through direct measurement. And then we can look at that a little bit. So that was the larger vision here, and thankfully, through the intrepid efforts of Max Planck, colleagues at Max Planck, and all the partners, Icarus is really a concept and a software solution as much as a hardware. So it's not so much, it's flexible with regards to the hardware. And Martin will speak about that. And the new way forward will be a microsat-based system. And I'll let Martin speak about that. But the larger, exciting dimension here is that whatever is being done here and having these receivers on space, it's already clear that further innovations have been and will further allow us to decrease this tag size as well as tag cost. And then you see how we are suddenly getting into this exponential part of the curve, right? That about the number of species that then the system would allow us to address, right? So with 3 grams, we're about here, but we would already be at 8,000 species. And here we were 5 grams, we already at 5,000. So now suddenly you push it by another gram, and you're getting to 60, 40 to 80% of species. And suddenly we're like, wow, opposite deployment is still going to be the key bottleneck, et cetera, but in principle, at least, the potential is there. And then I was talking about already about lynchals, about these tracks. What we haven't talked so much about is some of these overall products. Well, I talked about niches, and niche characterization as a product, right? But many of us already have seen these Brownian bridge-based movement corridors, migration corridors, and that's obviously an important spatial product. But other ones are, for example, home range, right? And here, AKD-based home range for that particular individual that was provided by data, Icarus data in this case, provided by Bayar, and here annotated with human modification data. And you see that that particular individual had its home range in a low impact area, like very low human modification. You can look at that at the animallives.org website. Same individual has this kind of EVI niche space, and you can flexibly look at all sorts of different variables. And then you can look here at this corridor. Here would be the or this particular flight migration route that it takes, supported with Brownian bridge models. This is where it had its home range, and then here we felt like we can say that the directional movement began in a way that allowed us to do these Brownian bridge models. And then this is where it ended up for a wintering range. We can also characterize the wintering range as a product. And then here, we're looking at the environmental niche that these points tell us about in between, right? Here would be the EVI. So it was mostly in very green areas. Down here, it had to stop over in very low productivity areas. That's a low productivity region. We're able to link this information in. Here, the global human modification. So it started out in very not much human modified place, but then it had to go through very modified locations. Till it finally ended up again in a low human modified place, where it then established its wintering range. Here the EVI signal, and here the and that's in December 21. Here low human modification in that place. This would be the home range incident. So what we've begun to develop are pipelines and Scott will talk a bit more about that. So this is Scott's work, other people's work here. So John Locher, AJ Raniketa, others have been contributing to this type of visualization. We now have Kagidan driving forward some of this UI user interface work and visual characterization. It's a very powerful way to begin to understand the multiple dimensions that this type of data offers. I encourage you to poke around animallives.org, where we have 5-6 species and a few dozen individuals available, just to sort of showcase what is possible. But the key point here is that you're able to now, through pipelines, automatically derive these products, home range size, movement paths in different forms, as well as these niches over time. And maybe I will leave it there. We've begun to also prototype this idea of like a 100,000 sentinels project with 1,000 cranes project. So this is through collaborators that the Max Planck Institute has been pulling together that allowed us to work with the existing GSM, in this case, tag data for many species of cranes, and begin to think about a global multi-species system. So that was the ambition here, right? To go away from, okay, there's historic, that's awesome, but then can we take that to multiple species? And cranes are an interesting system in that regard, right? And obviously highly threatened system, highly threatened species, and here just some initial results. And maybe I leave it there, and just thank you for your time. Thanks.